My name is Jessica Rosen, and it is March 4th, 2025, and this is my presentation on hemolytic anemia. Hemolytic anemia is a condition where red blood cells are destroyed at a rate quicker than the bone marrow is able to produce them. Red blood cells contain hemoglobin, which is an iron-containing protein that is able to reversibly bind to oxygen. Around 98% of the oxygen circulating throughout the body is bound to red blood cells in this way. When the body does not have enough red blood cells, or if the red blood cells are being destroyed at an excessive rate, nearly every organ and organ system can be negatively impacted because not enough oxygen is being delivered to them. There are a number of different forms of hemolytic anemia, as well as many different causes. For the sake of time, I will only be focusing on a handful of different types of hemolytic anemia, but please be aware that there are many more types that I will not cover in the next several minutes. Some forms of hemolytic anemia are caused by red blood cell abnormalities arising from the red blood cells themselves. These can be due to enzymatic issues, hemoglobin solubility issues, or membrane structure issues. Other forms of hemolytic anemia are caused by outside factors that end up affecting red blood cell structure, such as immune system attacking red blood cells or certain pathogens. Hemolytic anemias can also be categorized as being inherited or acquired. This means that some forms of the disease can be passed down from parent to child. So, abnormal blood work is generally a good indicator that a person has anemia. One indicator of anemia on a complete blood count, or CBC, is a decrease in hemoglobin, which is the iron-carrying protein in the blood. Another indicator of anemia is a decrease in the number of red blood cells that are circulating. Once anemia is diagnosed, additional tests can indicate whether or not the anemia is due to hemolysis. People with hemolytic anemia tend to have an increase in the number of reticulocytes on a complete blood count. Reticulocytes are immature red blood cells, and the presence of an excess number of them indicates that the bone marrow is producing additional red blood cells as the mature red blood cells are being destroyed. Examining the red blood cells via blood smear can be very informative when it comes to determining whether or not the present anemia is due to hemolysis or another reason. Depending on the irregularities the red blood cell shows, different types of hemolytic anemia can be diagnosed. There are several different forms of genetic hemolytic anemias that are caused by red blood cell membranes that are not structurally sound. One example of this type of anemia is hereditary spherocytosis. This condition can be caused by mutations to 5 different genes encoding for cell membrane structure, including ANK1 and EPB42. Hereditary spherocytosis is primarily passed down to the next generation through an autosomal dominant pattern. In other words, a person only needs one copy of the mutated gene to be afflicted with the disorder. However, because so many different genes are responsible for ensuring proper membrane structure, mutations to any number of them can cause membrane disorders of red blood cells. Because of this, hereditary spherocytosis can also be inherited by an autosomal recessive pattern, meaning a person needs 2 copies of the mutated gene to have the disorder. Normal, healthy red blood cells maintain their biconcave shape in part due to proteins between the cytoskeleton and lipid bilayer. People with hereditary spherocytosis have mutations to some of these proteins, making them less able to support the proper shape of the blood cell membrane. These altered red blood cells are smaller and rounder than healthy red blood cells and are called spherocytes. These cells are more prone to premature destruction. The presence of spherocytes on blood smears are an important diagnostic to narrow down the type of anemia a patient presents with. However, spherocytes can also be seen with immune-mediated anemia, so further testing needs to be done in order to rule out immune-mediated anemia. Family history is an important diagnostic as this condition is hereditary. Currently, there's no cure for hereditary spherocytosis, so treatment is based on improving symptoms and the quality of life for individuals with the disorder. Folate, a B vitamin, is often recommended for people with anemia as this is an important component of red blood cell formation. The treatment that is usually most effective for minimizing the negative effects of hereditary spherocytosis is splenectomy, because the spleen is the site of most red blood cell destruction. There are, however, a number of other risk factors associated with removing the spleen. Although no cure is on the horizon, there is still a great deal of research being done on the condition, as well as improving current means of testing for hereditary spherocytosis. One promising diagnostic method was recently studied using DNA sequencing to detect mutations in the genes responsible for hereditary spherocytosis. This study showed that DNA sequencing was highly effective at diagnosing the condition. Sickle cell disease is another genetic form of hemolytic anemia caused instead by changes to hemoglobin solubility. Sickle cell anemia is inherited in a recessive pattern and is seen most commonly in West Africa. Sickle cell disease is the result of a mutation on chromosome 11 to the hemoglobin beta gene, which leads to the production of an altered form of hemoglobin called hemoglobin S. Hemoglobin S, which is carried by red blood cells, can make the red blood cells stiffer, which leads to the classic sickle shape of red blood cells seen in sickle cell disease. These abnormally shaped red blood cells don't carry oxygen as efficiently and don't flow as smoothly through vessels, which can lead to blockages. The presence of sickle cells on a blood smear is the most effective way to diagnose sickle cell disease after confirming general anemia. Solubility tests are also used to determine whether hemoglobin S is present. One of the primary treatment options for sickle cell disease is the administration of blood transfusions. Blood transfusions increase the number of healthy red blood cells circulating throughout the patient's bloodstream, allowing more oxygen to be carried and reducing the risks associated with sickle cell disease. Another well-established treatment for sickle cell disease is the administration of hydroxyurea, a medication that increases the production of fetal hemoglobin, which in turn prevents the formation of sickle cells. Gene therapy for sickle cell disease is more recent development towards a cure for the disease. Gene therapy involves modifying the genetic sequence of hematopoietic stem cells by either adding a specific gene into a patient's DNA or by editing specific DNA sequences within a gene. Hematopoietic stem cells are found in the bone marrow and are responsible for producing cells, including the affected red blood cells of sickle cell patients. By editing these stem cells, they are able to produce healthy red blood cells instead of sickle cells. Gene therapy has been used successfully for treatment of sickle cell disease. However, there is still research being done in this area of study. Other non-genetic factors can also cause hemolytic anemia, such as immune-mediated anemia and those caused by toxins. Autoimmune hemolytic anemia is caused by the body's immune system destroying its own red blood cells. This type of anemia can be triggered by certain medications, blood transfusions, infections, etc. Red blood cells naturally contain antigens on the cell surface. In normal, healthy persons, the immune system recognizes these antigens as self-antigens and does not attack the cells containing them. However, in a person with autoimmune hemolytic anemia, their immune system does not recognize these self-antigens and instead attacks them as if they were outside invaders, leading to red blood cell destruction by phagocytosis. A specific test that can determine autoimmune hemolytic anemia is called a Coombs test, also known as a direct antiglobulin test. This test determines whether or not there are antibodies attacking a person's own red blood cells. A positive test shows clumping of red blood cells, called agglutination, and can confirm the diagnosis of autoimmune hemolytic anemia. For autoimmune hemolytic anemia, corticosteroids are administered, which suppress the immune system enough to hopefully prevent a person's own antibodies from attacking their own red blood cells. If this does not work, splenectomy is offered as a next step for some patients, but because the spleen plays a major role in autoimmunity, its removal can reduce the destruction of red blood cells. However, there are many potential side effects and risks associated with this surgery, so in recent years it has become less recommended. Although no cure is on the horizon, multiple different forms of treatment are currently being studied to treat autoimmune hemolytic anemia. One of these treatments has been shown some promising results is a medication that inhibits the activity of spleen tyrosine kinase, which plays a role in phagocytosis of cells. By reducing the activity of spleen tyrosine kinase, red blood cells targeted for destruction are less likely to actually be destroyed, leading to an increase in hemoglobin concentration. In conclusion, hemolytic anemias encompass a wide range of different conditions, only a small portion of which I have touched on in today's presentation. A great deal of research is still being done to discover new treatments and cures for hemolytic anemia, and as medical advancements continue to progress, hopefully we will see a cure for some of these conditions in our lifetime. Thank you for listening, and here are a list of references.